might still be some people dropping in. So, hi everyone. Welcome to our very first Motivated Cognition meeting. We are very excited to kick off this year's session with our guest, Brian Knutson. Brian Knutson is a professor of psychology and neuroscience. For those of you who don't know him, at Stanford University, where he directs the sympathetic and affective neuroscience lab, and where he has also completed his PhD in experimental psychology. His research and his lab uses a variety of methods, including self-report measurement, neuroimaging, psychophysiological assessment, and pharmacological methods, just to name a few, in order to investigate the neural mechanisms that underlie emotional experience, and also to explore the implications of this work for clinical disorders, disorders of affect, and also for economic decision making. And so in today's talk, he will present work on deconstructing neural predictors of risky choice. And as usual, we're going to have a 40 to 45 minutes talk, and afterwards we have plenty of room for discussion. So please join me in welcoming Brian Knutson. We're very happy to have you here today, and we are very much looking forward to learn more about your work.
Thank you so much, Zita. As I was saying, it's really a pleasure and honor to be here. I know like over half of you, it feels like. And so I hope there will be many more opportunities like this. And I appreciate the Motivated Cognition Network for putting all this together. I've really enjoyed all the other talks online. Today I'm going to talk about some work that has been really been evolving over the last 20 years or so in our lab. The most recent person to spearhead this effort has been Layli Mortazavi, who I'll mention later. And she has just published a paper on this, which I provided the link to that too. So if you're interested in a deeper dive, you can check that out. I'm going to describe some of the work here. But I also am tempted to add some other surprise data on rats. And so we'll see if I have time to get to that. But I couldn't resist. So I'm going to talk about deconstructing neural predictors of risk today. Let's see. People often wonder, what do we mean by risky choice? And in fact, many different people mean many different things, as our colleagues at Basel have shown. But when I'm speaking about risky choice, what I mean is a choice between a high variance option and a low variance option. This is kind of the standard economic or finance definition. So this is exemplified by maybe some of you are old enough to have seen this movie, The Matrix. In The Matrix, the protagonist, who's a hacker named Neo, played by Keanu Reeves, is offered a choice between a red pill and a blue pill by Morpheus, a sort of shadowy figure from another world inside the computer. And Morpheus basically says, if you choose the red hole, we don't know what's going to happen. But if you choose the blue pill, you go back into your world and you stay safe and sound. And of course, there probably wouldn't be a movie if Morpheus didn't choose the pill, which represents the risky option. And this kind of exemplifies the dilemma, which is if you choose the risky option, great things, really interesting things may happen, but also terrible things may happen too. So it's a high variance proposition, whereas the blue pill is a lower variance proposition. Maybe it'll be kind of okay, but you'll be back in your boring hacker life. Okay, so it turns out that risky choice is probably very ancient and probably antedates humans. So here's an example of a risky choice. It may not look like it because this gazelle is drinking water, but it turns out that it's actually tense because there's a danger lurking beneath the water. So even though it wants the water and that's a good outcome, a very bad outcome might also ensue. And so it has to be ready to leap away at a moment's notice. And so notice that this is happening on the order of seconds. And so what this implies about risky choice is that the basic neural machinery that supports risky choice is probably conserved not just across humans, but even across vertebrates. And this is a diagram that was in a publication by Paul Chizek where he sort of tries to trace back different parts of the brain and when they evolved and what their function was. And he traces back all the way to the emergence of the vertebrates, this sort of visually guided approach, reinforcement learning, meaning that you can learn about good things, but you can also learn about bad things and sort of switch your choices around. So this implies the existence of some kinds of machinery that might support risky choice at a moment's notice. And even before the vertebrates, you have the hypothalamus, which is involved in moving forward, and so forth. So this is just to illustrate that probably the basic components that support risky choice are ancient evolutionarily, and that implies that they probably are not seated in the cortex, at least in the prefrontal cortex, even though that probably plays a supporting role. That probably some of these components are subcortical that will support risky choice. Um, let's see, so. But the human study of risky choice, of course, has occurred much later in evolutionary history in, well, near Zurich. Um, uh, so the first place where the formal study of risky choice is documented is in the 1600s in France, where Pascal and de Fermat uh, corresponded with a series of letters to try to determine how to help an Italian nobleman value his bets when he left the table. And their innovation was not that we should not just consider potential good and bad outcomes, but we should also consider the probability with which those outcomes will occur. And that's been called expected value. And that's a core foundational concept of much of psychology and also of economics. That is an attempt to quantify the value of a risky choice. This is Pascal. I don't know if he actually said this, but I like the quote. The excitement that a gambler feels when making a bet is equal to the amount he might win times the probability of winning it. I like the excitement part of the quote, so that's why I'm not sure if he said it. But anyways, I was able to identify this in some literature. If somebody can find this source, then please let me know. Um, okay, so and since then we've been thinking about risky choice, and not just academics, but also the casinos of Las Vegas and insurance companies and all actuaries of all kinds have been using this formula to evaluate risk and quantify it and monetize it in some cases. So this is kind of again an operationalization of what we mean when we're talking about risky choice. So today, what I'd like to do is just talk about some research which attempts to identify circuits in the brain, in the human brain, that actually predict that you're going to take a risky choice or not, using gambles, actually. So harkening back to Pascal and de Fermat, and then ask the question, can we manipulate the activity of those circuits and actually control risky choice? And then finally ask the question, well, do these results generalize outside of the laboratory, at least for an individual? So the way that we think about studying this in my laboratory is it has sort of two parts. One part is we're interested in when stimuli go into the brain and cause correlated activation, are there brain circuits that anticipate good and bad outcomes, basically, um, which would be a precondition for making a risky choice, according to Pascal. The second, and we call that an input model, and that's sort of a basic question of affective neuroscience. If indeed these anticipations of good and bad outcomes actually cause affective experience too. The second question is what we call more of an output model, which is, does the activity in those circuits have the capacity to influence choice, and in this case, risky choice? And that's a central question of neuroeconomics as well. And so you can think of this, this very simple diagram of a brain below where there's input coming in time. It causes correlated activity. That overlaps with predictive activity, which then generates output. And then, so we're kind of interested in this intersection of the input and the output. And you can even think of these as different circuits. And you can even think of this diagram. This actually describes our research over the span of a couple of decades. The first we see input question, and then we move to the output question. But also, our experiments are set up this way, where we'll present input to a subject over the course of seconds and look for correlated activity, and then look for predictive activity that predicts the output. And this can happen, in 6 to 8 seconds, for instance. Okay, so where would we look in the brain? Well, I already hinted to you that we think it's not going to be sufficient to just to look at cortical activity. We're going to need to get under the cortex. And there are other reasons that are more recent in scientific history to think this. One is the phenomenon of self-stimulation, which was accidentally discovered by Olds and Milner in Canada in the 1950s, in which they were able to show that rats will work very hard to be able to self-stimulate certain circuits of their brains. And these circuits tend to lie along the midline of the brain and deep in the brain, running up from the midbrain to the ventral striatum and also to the medial prefrontal cortex, which coincidentally was later found to house dopamine receptors and to receive dopamine projections from the midbrain. So we think we're going to need to target that circuitry, and indeed, much work has been done since that time, not just in humans, but also in tracing the tracts of this circuitry in monkeys and showing that yes, dopamine does project from the midbrain up into the striatum in a sort of a beautiful kind of ascending trajectory, going from the medial part of the brain to the lateral part of the brain, and then sort of extending along the striatum in that direction, and also to the prefrontal cortex. But then there are reentrant projections that come back from the prefrontal cortex into parts of the striatum, through the globus pallidus and the thalamus, and back out into the prefrontal cortex in a so-called ascending spiral kind of looping pattern that seems to project from the ventral medial parts to the dorsolateral parts. So Mogensen and others have described this as a potential motivation to action interface, so that dopamine in the ventral medial parts of the circuit can promote action. I'm sorry, motivation. And as the projections move dorsolaterally, they actually promote action. So this is kind of where our target area, one target area, these mesolimbic dopamine projection regions of the brain, some of which are subcortical. And then because I am talking about neuroscience, I'm contractually obligated to show the Wolfram-Schultz slide, which shows that dopamine not only responds to reward, so when the monkey thinks it's going to get juice, and it gets the juice, then the neurons fire, didn't expect to get the juice. But once you cue the monkey that, yes, you might get juice, then the firing of the dopamine neurons moves to the presentation of the reward cue rather than delivery of the juice. And when you deceive the monkey and present the reward cue and don't present the juice, then it actually physically stops firing here, indicating that it's not just a surprise signal, but it seems to be signaling something about the anticipation of reward, not just getting a reward, but anticipating getting a reward, which has been called a prediction error in reinforcement learning terms. Okay, so this is just more evidence that yes, we know that this circuit probably is somehow involved in motivated choice, at least, and possibly in risky choice. But we don't know like which of these projections is really critical. And this is where functional magnetic resonance imaging comes along in 1992 and gives us a window into the brain, not only at the second level, but also at the couple millimeters level and subcortically, so that we can actually ask this question in time. So if you go back to the Schultz slide, which I'm not doing. Whoops, sorry, I'm going forward, not back. Well, I just have to re-describe the Schultz slide. That's on the order of seconds, right? So we need to get, with our neuroimaging modality, not just visualization at the time of the outcome, but before the outcome occurs, right? And so we and others have used video games where people try to make money or avoid losing money to try to visualize what's going on in the brain, not only when people get rewards, or also when they lose money, not just gain money, but also lose money, but also when they're cued that they might get a reward or they might get a punishment. So we can then look at what's happening during this anticipatory interval, which is a couple of seconds maybe, and see, is something happening in the brain that is correlated with the anticipated value or the expected value of what the person might make by pressing a button rapidly to a speedy target. And of course, then you can manipulate aspects of the cue. So you can cue the subject that, yes, you might gain money or you might lose money. And you can put lines in the cue and indicate, more is at stake or less is at stake, and so forth. And so I won't belabor this research, which there's a lot of it now, much more than I ever anticipated. other than to say that, there's a sort of a consistent set of results that we can get out of it. This is like the input question that I mentioned, where we see that when people are anticipating making money, we often see increasing activation depending on how much they think they can make in this one region that I was referring to, the ventral striatum or the nucleus accumbens. So the more money you think you can make, the more activity we see there. And it tends to correlate with how excited people say they feel, so how positive and how aroused. Um, there are other regions like the anterior insula, which is just adjacent to it right here. Oh, I'm sorry, I'm not doing my little uh, designation of where in the brain we are. So this is a slice of brain, like if you cut my head here and took off my face, like in Westworld, and looked into my brain, then you would be looking at this coronal slice of the brain that I'm showing right here. And the idea is that this ventral striatal region is indeed one of these medial regions that the midbrain dopamine neurons project to, including the nucleus accumbens. And this adjacent old cortical region, the insula, anterior insula, not only responds to anticipated gains, but also to anticipated losses. So it looks more like kind of an arousal signal. And then finally, the medial prefrontal cortex also responds to anticipated gains and as well as the probability of those gains. So we have the reason I'm showing you this is because then that gives us targets, input targets that we can then use to try to predict the output, which would be the choice if they indeed are involved in choice. And so when we think about risky choice, again, going back to Neo and the example from the Matrix, then risks involve uncertain gains and losses. Um, and so they probably should activate both of these regions, but the balance of activity in these regions will differ with respect to whether you're going to choose to take the risk or avoid taking the risk. And so the idea is that the nucleus accumbens activity should predict that you're more likely to take the risky choice. And the anterior insula activity, in combination with the nucleus accumbens activity, should predict that you're actually more likely to avoid taking the choice. Alone, it's going to just signal arousal, but if you put them together, it may show you a slightly different signal. This is, by the way, all with respect to fMRI activity, which is sort of oxygenation in the brain. Many of you are familiar with this, but not everyone. And so we can only make so many inferences based on the changes in fMRI activity that we see. But we're inferring that there's more activity in that region of the brain, basically. Okay, now this is a little bit of a side trip for the imaging geeks here. Um, you might say at this point that you shouldn't do this. You shouldn't be asking these questions with fMRI. Why? Well, and these are recent criticisms, or at least recent for me, criticisms of fMRI. One is that, well, actually, you probably aren't going to be able to localize separately any affective or emotional activity in the brain. And this criticism actually goes all the way back to William James in his 1884 article, What is an Emotion? And I won't belabor it, but James actually makes a very clear distinction between two hypotheses, one in which emotion is represented by sensory motor cortices, basically, and another in which it's represented by some other region that's localizable, but it's not the sensory motor cortices. And he falls squarely on the sensory motor side. And this is reiterated by a more recent review by probably some of you out there, where they sort of conclude that yes, it's really you shouldn't be able to actually localize a separate kind of substrate that is representing affect or emotion. And they actually quote James to their credit. So there's a real question here whether we're going to be able to actually dissociate substrates that are associated with, say, what I would call affect from these sensory motor activations. A second set of questions have to do with reliability, and these questions have to do with maybe we won't be able to localize anything because if we use different ways of analyzing the data, then we'll find different results. And in fact, this team actually analyzed a large data set of risky choice data on a gambling task to demonstrate this and published it in Nature. So this is a real question, like, will we actually be able to use different modes of analyses to show the same thing? And finally, a more recent criticism is that, and you can read it right here, this is their conclusion: reproducible brain-wide association studies, meaning with behavior, require thousands of individuals. So the idea here is that the effect sizes are so small that you're just going to have to get thousands and thousands of individuals. Now, to be fair, they're not looking at risky choice. They're looking at sort of activation over the entire brain and some kind of vaguely defined psychopathology variable. So it's a different kind of a question they're getting at, but this is a pretty strong claim, and it's one that I think that we should take seriously. So, and I'm going to claim that I'm going to address all of these today. All right, so we'll see if you're convinced by that. Okay, back to the findings. Okay, but I want to give you more intuitions about what we're doing here. So basically, we're having people play a very simple gambling task, or at least what we think is simple. And so this is an example. So some of you might look at this gamble that I've just presented, and I would ask you, do you want to take that gamble or not? And I can't see all of you right now because I'm looking at my PowerPoint, but I would hazard to guess, based on many, many audiences I've spoken to, that almost nobody, maybe a few people, will take this gamble, right? However, if I present this gamble to you, then often things will change. Often a few people will take that gamble. Not many, but not a majority, usually about 20 to 30%, but some people will opt to take this gamble. Now, if I present a third gamble to you and ask how many people are willing to take that gamble, often over half of the audience will take this gamble. And there's one exception, and that is Swiss foreign exchange traders. And I don't know why they're different, but that's the only audience I've found where we don't observe this pattern. So maybe you want to study them in Zurich. Um, but at any rate, most audiences show this pattern in most countries I go to. Okay, so is that crazy? Well, yes and no. Because it turns out that if you calculate the expected value of each of these gambles, that is, if you play it over and over again, it is zero. Okay, they're all the same. And if you calculate the variance for each of these gambles, it's also going to be zero. Okay. Um, so if you're a finance trader, you're not going to distinguish between these gambles because they have the same expected value and the same variance. And yet people show this preference. Now, there are two questions here. One is, why do people even say yes in the first place? And the second is, why do they prefer this, what we call the positive skewed gamble? And this is just showing that, yes, from an expected value and risk standpoint, they're the same, but from a choice standpoint, they really aren't the same. And we often see this pattern where people prefer the positive skewed gamble. So this is kind of a puzzle. It's one of many puzzles in economic choice. And it's just to give you sort of a taste of the problem that we're faced with. The problem that we're faced with is both one of rational choice and of irrational choice. And here I use rational in a sort of a finance way, like a quantitative way. Okay. So this is the experiment that I'm going to be talking about. Well, one of the two experiments where people are faced with a choice between a high risk gamble and a low risk gamble. Those gambles can appear on the top or the bottom of the screen. They are for real money. They are evaluated on each trial. People get the outcome and it's added to their total, and they continue and make many other gambles. Then the paradigm also asks people to indicate with the left or right button press which a choice they want to make. The gamble is, by the way, always juxtaposed with a low risk option, which is certainty that you'll make $0. And so basically, these options are counterbalanced. So sometimes pushing the left button will give you the top option. Sometimes it will give you the bottom option, and so forth with the right button. And we're doing all this as people are being scanned with fMRI. Okay, so that's sort of the layout. By the way, if any of you have questions, just yell at me, and I will stop and try to answer the questions. Okay, so Lely Mortazavi looked at some data that we had collected, two data sets basically using this task. And she analyzed that data. This just shows the sort of choices of people, suggesting that they're kind of risk-seeking actually in these two samples. More than 50% on average are choosing to take the gamble, which is a little bit surprising. But she also analyzed two other data sets with a different gambling task that some of you may be familiar with. And this is the task from Tom et al. From their 2007 science paper, in which they have balanced gambles. So they don't have these sort of skewed gambles. We also have balanced gambles in our data set. And basically you say yes or no, I want to take that gamble with varying degrees of certainty. And what they manipulate is the potential gains and losses from those gambles. And so Lely analyzed basically the initial science data set, which was fairly small, about 15 subjects, and also the larger data set I mentioned, that was the 108 subject data set from the Nature paper that basically was claiming, okay, the way that you analyze the data is really going to make a huge difference. Okay. All right. So what what did she find? And the question here is, are there sort of reliable neural predictors of risky choice? And if so, where are they? And do they generalize? Okay, so here's just this is not fits. This is just looking in these volumes of interest that I mentioned. So this is the nucleus accumbens, this is the medial prefrontal cortex, and this is the anterior insula. These are based on, for instance, the mid-task. Um, and we can then extract activation and basically bin it as a function of you're going to take a risk or you're not. And what you see is a very strong signal in the nucleus accumbens, which indicates in the first two data sets, so this is the skewed gambling data set, that the person is going to take a risk. This white bar represents the period when they see the gambles, but before they've chosen. The yellow line is are the trials where they're going to take a risk. The gray line are the trials where they're not going to take a risk. So you see this in both the original and the replication data sets. You also see a not as strong signal in the medial prefrontal cortex, which indicates they may take a risk. And in the anterior insula, there's no discernible signal during this time, but you're going to see that when we use a multivariate model, then something pops out of here. Um, what about the Tom et al. Data set, and what about the Botvinnik-Nezer et al. Data set with the balanced gambles? Well, we see a similar pattern, basically. So here's the original Tom et al. Data set, where you see that the nucleus accumbens is again predicting you're going to take a risk. Medial prefrontal cortex also is again taking you're going to take a risk, and that the anterior insula is not showing univariate differences. And here in the second data set, it's kind of interesting. You see the same patterns, but it's much, much smaller, right? So the effect size is smaller here. Um, and you might be wondering why, as we were, but we had a suspicion about why. And to just cut to the chase, when they collected this data, they used what's called a multi-band pulse sequence for acquiring their data. And when you use that pulse sequence, and I won't go into the details, but those interested should definitely email me. You induce high frequency noise into the center of the brain, which happens to be where the nucleus accumbens is, and also in part where the medial prefrontal cortex is. And so, unfortunately, blunt signals that you might see there. And this even works with a phantom. It's not just, dependent on brains. So we unfortunately had to do a lot of work during the pandemic to figure this out and publish it, but we now have evidence that this is the case. So you can replicate in this data set. It doesn't look as good because there's basically more noise in the signal in these regions of the brain. So again, a side note for you fMRI geeks out there. Um, okay, so now we can do a multivariate analysis. So we take the activation out of these regions, we put them in a multiple regression, we try to predict risky choice, and we see across these data sets again, you have quite a strong coefficient for the nucleus accumbens, and you have a weaker coefficient for the medial prefrontal cortex in most data sets, um, and you have a negative coefficient for the anterior insula across these data sets as well. Okay, so you're getting additional information from the anterior insula, not in the same direction. If you do a signal detection analysis using the activation from these regions, you get pretty decent areas under the curve of about 0.76, 0.78, and so forth. It's a little lower again in this the Botvinnik-Nezer et al. Data set. It's about 0.72. So it looks like this pattern is replicating. Now, what about just sort of theory-free analysis? So this is a highly specified analysis in space and time. It's sort of theory-driven. What if we just throw all the data, all the gray matter data, into a classifier, and we classify, are you going to take a risk or not? Do we recover a similar result? Or maybe James is right. Maybe all of the action is up in the motor cortex and the visual cortex, right? It could be. It could be. So let's look at that. But what we see when we do that is a very similar result. That the features that are extracted by a support vector machine with recursive feature elimination are positive in the ventral striatum, including the nucleus accumbens, and negative in the anterior insula, right? So this is not theory driven. So this is a different kind of analysis producing basically a similar result. And by the way, the classification indices are not that much better than just using the volumes of interest. Okay, so we're starting to answer the questions I raised, at least for these data sets. Um, What about let's really dig into this James idea, right? That that everything is distributed versus you can localize an anticipatory signal that predicts risky choice. Now, you might remember that we counterbalanced basically the sensory properties of the presentation of the gambles. So we presented the risky gamble on the top or the bottom. So we know that there are parts of the visual cortex that respond to that, different hemifields in the visual space. And we also counterbalance the left or right button press response. So if we're using classifiers at each of these stages, I just showed you this stage right here, then we should expect a different, if we can localize and dissociate this activity, we should expect different results for the input and the output and what's predicting the risky choice, basically. Can we do that? And the answer is yes. We can do a triple dissociation, basically, where when we do the classifier on the sensory input, we see this nice discrimination in the visual cortex, but not in the nucleus accumbens and anterior insula. When we look at the anticipation of choosing a risk versus not, we see this nice discrimination that I just showed you in the ventral striatum and the anterior insula. And when we look at the output, that is, are you going to choose a right or left button press? We see a beautiful discrimination here in the motor cortex, but not again in these other areas that I mentioned. So just blowing that up, you can see that there's basically a triple dissociation where we can dissociate input from this anticipation from output. So this is my favorite. If you don't remember anything else, remember this, okay? Because James is wrong. Sorry, James. But I honor his clear operationalizable prediction. I think that's great. You got to give him credit. And he writes way better than I write. Let me tell you. I'm so jealous of that. Okay. All right. So we talked about predicting choice within a subject, right? But what about across subjects? Subjects show individual differences and how much they prefer to take the risk versus not. And, if I had been able to pull you, we would have seen individual differences, right? So, can we say something about those based on the activity and of interest? And the answer is yes, to a certain extent. So we don't see, much to my surprise, across the gambles, if people are opting to choose the risky option, we don't see much of a discrimination in the nucleus accumbens, but we do see that the medial prefrontal cortex positively and the anterior insula activity negatively seems to be related to preference to take risk versus not to take risk overall. Now, if we look at the specific gambles, we see a similar but slightly different pattern. And that is that when we simply look at the positive skew gamble, your preference for that positive skew gamble, we see quite a strong relationship. And we have replicated this in several data sets now between what's happening in your nucleus accumbens and your seduction by the positive skewed gamble, basically. So those people who really want to take that positive skewed gamble tend to show a much bigger nucleus accumbens activity when they're considering that gamble. Now we see similar patterns for the balanced gamble in the medial prefrontal cortex, and this opposite pattern again for the negative skewed gamble in the anterior insula. So these are the more fine-grained predictions that we would have. But we do seem to see some activity that's lining up with, at least in the lab, with the tendency to choose to take a risk on these different types of gambles. Okay, so what can we say about these ideas? Well, based on this data set, well, I believe this is good news. So we can say that at least when we set things up right in the experiment, and we can dissociate the different stages of processing, we can dissociate out anticipation of taking a risky choice from the other kinds of sensory and motor activation. Um, we can also say that these results are pretty reliable. They seem to work across different tasks, different data sets, different analyses, and so forth. So at least with this kind of a paradigm, this kind of a Pascalian paradigm, they seem to generalize. And we can also say that they're pretty robust. I didn't really dwell on this, but the kinds of Z values that we get using nucleus accumbens activation to predict risky choice within subject are on the order of 5 to 7. So like these are huge, robust statistical results. Now, your results will vary as a function of many variables, including like how well delineated in time is your paradigm? Are you using multi-band versus single band acquisition? Did you control for motion? All this boring stuff that you don't want to hear about in this talk, but that we kind of obsess over, right? But you can achieve pretty powerful results within subject at least with these techniques. So I think that within constraints, they're robust. Okay, and if you could have just asked Neurosynth too. So you could just go to this huge database that has all these brain imaging studies, and it basically associates words and abstracts with maps of activation. And you can type in the words and you can say, show me what's associated with this word appearing in the abstract. And so in this case, these are a few studies because these are sort of finance studies. And this was done a while ago, actually now. If you type in the word mean, so meaning like, the expected value, you get this kind of nice activation in the ventral striatum, including the nucleus accumbens. But if you type in the word variance, you tend to see something different in the anterior insula. Let's type in different words like reward and pain. So if you type in the word reward, again, you see this nice ventral striatal activation. This is more studies, obviously, over 500. If you type in the word pain, you see kind of a different pattern of activation, again, in the anterior insula, anterior cingulate. And you can even look at, Randy Buckner was nice enough to put 1,000-person functional connectivity data set in there, which means you're just lying there and you're just looking at what's correlated with what in the brain. And you can see different regions. So if you seed the nucleus accumbens, then interestingly, you see negatively correlated activity in the anterior insula. If you see the anterior insula, you see negatively correlated activation in the nucleus accumbens. And this is kind of interesting from that standpoint of, when you present people with a risky choice, you're activating both of these regions, but they have different implications for the next behavior that you're going to show. Okay, so what can we conclude from this? Boy, I really should have a watch here because I have the feeling I'm going over time. Um, okay, here's what I'd like to claim. Neural circuits are activated in individuals before risky choice. Some of them predict that you're going to take the risky choice. Others predict that you're not going to take the risky choice. This is fMRI activity in humans. The neural circuits predicting the risky choice can be triply dissociated from those associated with sensory input and motor output. Oh, I'm only the second part of the talk, and I think I'm like out of time. Kendra, can I ask you, how much time do I have?
You're at almost 40 minutes.
Okay,
So
I'm sorry, I'm gonna have to blast through the rest of this. I was gonna talk about optogenetics and rats, but I'm not going to. But if you're interested in that, email me and I will send you a paper. Um, but I will talk about one thing, which is like, okay, what if we tweak these circuits, right? Can we actually perturb? Is this causal? Can we perturb risky choice? I'll show you the fMRI example of this, which is kind of corny, but I'll just show it anyway. So we did this a while ago. We showed heterosexual males three types of pictures, and then they asked them to take a risky choice. Again, a high variance versus low variance choice. So we showed them erotic pictures, we showed them snakes and spiders, or we showed them office supplies. And then we asked them to take these choices. And the question is, do they take more risk after seeing the pictures that are supposed to activate the nucleus accumbens and make them excited? And the answer is yes, they do to varying degrees. Not after they see the snakes and spiders pictures, not after they see the office supplies. Um, the other question, of course, is we were scanning them while they were doing this. So is the nucleus accumbens both responding to the pictures and predicting that they'll take this 50-50 chance of winning the $1 versus 50-50 chance of winning the 10 cents? And the answer is partially. So if we look at nucleus accumbens activity, it is increased by seeing the erotic photos to varying degrees in these people. And that then predicts that they're going to choose the risky choice. And so you can get a partial mediation by the nucleus accumbens activity of this effect. So this is just at least psychological evidence that's consistent with an idea that like when you start perturbing these circuits with irrelevant stimuli, you might be able to move around the risky choice. Okay, this is all about rats trying to optogenetically manipulate their nucleus accumbens, trying to perturb their risky choice. It turns out you can do it. It's crazy in science fiction. I don't have time to talk about it, but it's pretty cool. We did this in combination with Diceroth Lab, and Kelly Zalakuski headed that experiment. But the one point to take away from this is activating the nucleus accumbens can increase risk seeking, even when it's by stimuli that are irrelevant to the gamble. And then the extension that I'll show you is so one idea is that if you show this pattern often enough, it might accumulate and include and influence your life financial outcomes, which is kind of crazy. But so the idea is that if you anticipate gain enough, you might accumulate more assets. If you anticipate loss enough, you might avoid more debt. And so Laley actually looked at this in our data sets. We were able to collect measures of assets and debts in these subjects. So this is an extension of the individual difference idea. We did not see an association between nucleus accumbens activity during the positive skew, in anticipation of the positive skew gamble and assets, but we did see a negative association between activity in the anterior insula activity while anticipating negative skew gambles and debt as predicted. And this was actually after controlling for lots of other variables like sex, education, their actual behavior, their stated risk aversion, and their sort of a financial measure of risk aversion. We have replicated this result, or I should correct myself and say Laley has replicated this result in a different data set. So I'm starting to believe it, even though it's kind of crazy. Okay, so the point I'm just trying to make here is that it's interesting to think long term outside the lab. Could these neural predictors of risky choice apply to life financial outcomes like assets and debt? And it's an interesting question that is worth asking. All right, so you've already heard this. We can predict risky choice in individuals. We can control it on a single trial basis, at least in rats. And these neural predictors of risky choice may foreshadow life outcomes. So there are many implications to this, which I won't delve into because I think I'm running out of time. But many questions arise, like, are people aware of this? If so, can they change it? On what basis? Like, is it a chronic change or something they'd have to exert right in the moment? Another way of approaching this is like, if there are individual differences in these tendencies, could we change them through choice architecture or even through prostheses, even through software, that helps you to make choices? Another area that I wanted to talk about, but that will have to be another talk, is neural forecasting. So this is the idea of using the brain activity of a small group of individuals to forecast demand out of sample in markets for various products and propositions, which is another active area of research in our lab. Theoretically, I think these are really interesting results. I tried to make the case. One general idea is that we're trying to move from the input model to the output model, from description and explanation to prediction and control. And to improve, I hope this can eventually improve theoretical models of decision making with neural constraints. And one idea we obsess over in the lab is linking levels of analysis. So going quantitatively from these different one level of analysis to another. So for instance, going from the neurochemical level to the sort of anatomical level to the experiential level, and then finally to the behavioral level, and maybe even further to groups of people behaving, right? And so to really being serious about these mappings and stating what maps and what doesn't, and understanding like how we can have a replicable framework for making these predictions. So, what will we choose? Will we give up this research and believe William James and take the blue pill, or are we going to forge ahead and link these levels of analysis and do this crazy research? Well, part of it will depend upon funding, of course, but part of it will depend upon enthusiasm and openness. So I am habitually inclined towards the red pill, I have to admit, unfortunately or fortunately. So this is of course, the most important slide where I acknowledge the people who did the work. Laley did much of this work on the risky choice, which spanned over 10 years. She will be on the market soon. She also is working with rats and has all this amazing research in collaboration with the Dice-Roth Lab. So check her out. Kelly Zalkuski did the work with the Dice-Roth Lab that I didn't get to. We have strange sources of funding that tolerate our weird ideas. And so we're very grateful to them, some in Stanford, including the NeuroChoice Initiative. Thanks to you all across the world in different time zones for tuning in, and especially I'm looking forward to your questions. So thanks for being here. The end.
Well, thanks so much, dear Brian, for presenting this fascinating research. We have now about 15 minutes for discussion, questions. So just raise your hand if you like, and I'll just call you. Or alternatively, you can also type into the chat and I'll read out your questions. Yeah, Kendra.
So Brian, I was really struck by the last part where you were talking about the anterior insula predicting debt, because I don't know if you remember this, we also found that the anterior insula was predicting people's likelihood of getting defrauded in one of our the data set we published a long time ago.
Oh
So I'm wondering,
yeah.
it's
Yeah.
very deep in the
Wow.
supplement of one of our
Yeah,
papers.
yeah, I know we shoehorn these little cool
Yeah,
facts into the supplements. We do that
yeah.
too. Yeah. Yeah.
So I'm just curious, what other real life things do you think might be predicted by anterior insula activity? Like, what other kinds of things are you thinking of looking for? What else is on the horizon, perhaps?
So I can just refer to other literature. So um I'm trying to remember the first author's name. It's not good. But work that Cami Cunin did with the Bond Group suggested that if you look at investors and you look at their portfolios, actually anterior insula activity in a gambling task is related to some measure of risk aversion in their portfolios in Germany. So that really struck me. It was kind of published in scientific reports. I thought it was undervalued, but it seemed to be a very interesting result. So I think this is worth pursuing. And I didn't know about your results, so I'm glad to hear about it now. So I can sort of put it in that portfolio of, whoa, this is interesting and cool. I think part of the challenge, and there are probably others I don't know about that you could tell me about right now if you want. Okay. Well, I think it's worth kind of tracking this stuff because there's not an obvious funding agency. We don't have a National Institute of Wealth here in the United States. We may not even have a National Institute of Health soon. But we don't really have dedicated funding agencies to looking at this. But I think life financial outcomes are understudied and studyable and quantifiable. And there are also interesting cultural differences too in financial behavior that are worth looking at. Um, so I think it's like an amazing opportunity, right, to quantify reliable life financial outcomes. And I think part of the challenge we've been faced with at least the neuroimaging, is just getting a reliable signal out of the paradigms that we can use. It's because you have to have reliability. Lorene's here, Renato's here. They know you have to have reliability on both ends, right, with the neural signal and with the phenotypic measures and with whatever the life financial outcomes are. And so there's a lot we don't know about, like selecting good measures that will maximize our chances of finding links. But the fact that there are these links suggests to me there's something really there that accumulates over the course of a life.
Right. Thank you. Any more questions? Then maybe yeah, Lorene.
Hi, Brian. That was cool. Thank you.
Good to see you.
Hey, this is an obvious question probably coming from me, but do you have diffusion data for these guys?
Yes, I wanted to talk about diffusion data, but like I ran out of time. Okay, so like, thanks for asking. I should pay you $20 right now. Um, so just to signpost this for you guys, um, we are interested in multimodal data. Diffusion weighted imaging is a way of looking at, where is the water moving as a proxy for something about like the coherence of the myelination of tracks that connect one neural region to another. Um, and so what we're starting to find is we can track diffusion along tracks that connect to the nucleus accumbens. We jokingly call it the connectome. Lorene published a really cool paper on this a few years ago in PNAS. You should check it out. It's more on relapse to addiction, but what we're finding is we can get these reliable sort of metrics of diffusion along these tracks, and that some of those, the diffusion along some of those tracks is related to interesting psychological and financial variables. One of them that keeps popping up, and I think Renato was on a paper that's related to this as well, is this track that connects the midbrain to the back of the nucleus accumbens. And that diffusion along that tract seems to be related to impulsivity variables like you would measure with the Barrett impulsivity scale, which obviously has implications for addiction. Other tracks that go into the nucleus accumbens are coming from like the prefrontal cortex or from the anterior insula. We can track diffusion along those tracts. Those seem to not be correlated with this impulsivity variable. They do seem to be correlated with relapse. Lorena has shown in stimulant users. They may be correlated with other really interesting variables. And just to promote Laylee's work a bit more, she is actually looking at these tracks not only in humans, but also in rats. And is able to actually use brain clearing to verify the existence of the tracts. and then and use optogenetics to control the output of the tracts and see, whether they have an appetitive or a aversive effect. So she's writing some of that up right now, and that's very exciting. That's jumping down a level to the physiological level. I think I've now strayed far from Laureen's question, which was probably about a specific anterior insula to nucleus accumbens projection. And is it related to any of this? And the answer is yes, it is. So Laley has data on this. It's related, but I'm not remembering the exact results, but I know it's related to brain activity when people are anticipating losses, for instance, in the anterior insula. And that is related to some of these other variables that we were talking about, like tendency to take risks and also maybe to debt in the long run.
We have a question by Fabio.
Oh, he disappeared.
Fabio, oh, okay. So then we'll move on with.
Hello?
Yeah, we can hear you. Yeah.
Okay, can you hear me now?
Yes.
Yes.
Okay, that's great. Thanks for the talk. It was very, very thought-provoking. I have a question on a bit more meta plane of thinking. So as I listened, I felt kind of reminded on the reinforcement sensitivity theory from Gray. Are you familiar with that one?
Yes. Yeah. I'm fairly familiar with it. Yeah.
Yeah, so I think there's quite an overlap between the behavioral activation system and the behavioral inhibition system regarding
Yes.
the neuronal circuits which are involved. So
Yes.
I wondered if you see any potential to perhaps develop better measurements for risk prediction in combination with the measurements already working for the reinforcement sensitivity theory, because as of now it's quite a hassle to find measurements which are reliable and valid to predict behavior. Yes, what do you think regarding
Yeah, I
that?
totally agree. And in fact, we owe a debt to that whole line of research, right? Starting with Pavlov, going to Eysenck, going to Gray, right? Thinking about these different motivational systems, measuring them sometimes with psychometrics. Often we will collect individual difference measures, including not only the NEO, but also the BIS-BAS scales, which are meant to tap into this behavioral inhibition, behavioral activation idea. They all correlate together as they should psychometrically. Also scales with positive arousal, negative arousal. So now one might say, okay, then the obvious thing to do, I think, from my perspective, is correlate that with these sort of brain measures we're seeing, or maybe the diffusion weighted imaging measures. And we have done that. We haven't found really strong. Let me correct that. We have found correlations actually. So if you use the mid task and you don't blow out the middle of your brain, and you correlate that with this kind of brain activation versus brain and behavioral activation versus behavioral inhibition scale, you see that especially the behavioral activation measures and extroversion for that matter tend to correlate with the nucleus accumbens response when anticipating a large reward like $5. And we've now replicated this after 10 years. And so again, we find these results in one data set. The order of the correlation is about 0.33. So it's not very strong, but it is kind of what we would expect given the reliability of the measures. The correlation of the behavioral inhibition slash neuroticism slash negative arousal with the anterior insula activity when people are anticipating large losses is something we anticipated and reported, but I don't know if we've replicated that. So Laley actually knows about that, and she's here. But I'm not sure if that's as strong. But I think this is a great approach, and it needs to be done. And we have new tools basically to see what's going on in these regions of the brain and their connections, and we should absolutely be using them.
We have a question by Sammy Walls.
All right, well, so thank you for the presentation. I also have a question about the neuro forecasting theory from
Yeah.
Alex, and then mainly because most of the research kind of draws on the fMRI literature, and I was wondering if you see a parallel with the neural synchrony measure in EEG, for example, or if you could elaborate on EEG
Oh,
visual.
wow. Yeah. Great questions. Yeah, as I mentioned, I could go on for a long time about neural forecasting. But just let me state one implication of this work, right? One implication of this work is that you could use these target activation from these targets not only to predict individual choice, but also in a group to try to forecast the choices of people that are not in the group, right? And that's what we call forecasting because it's out of sample. So within subject, we call that prediction. And that's what I talked about today. But out of the group would be forecasting. And it raises this interesting question of what is the best forecaster of group choice, right? Is it the actual choice of your group, which if you're a behaviorist or revealed preference theorist, you should think it's that. Um, or is it actually the brain activity, which would be counterintuitive, but it is a pattern that we have seen in some studies of neural forecasting. And that raises, if it is, then that raises the question, what is most generalizable about choice, right? The process of choosing something, if you break it down and measure the different stages, maybe some components of that process are more generalizable than others. And that's interesting from a choice theoretic standpoint. And I won't say that we nailed that down at all. We're sort of at the stage of like, well, let's figure out when the brain activity adds value to the behavioral forecasts, when it actually is the only thing that forecasts, why that is. So we're at a very early stage, so I'm not giving you a satisfactory answer, but it's a great question that has emerged from these findings in this literature that we did not anticipate actually. So yeah, so stay tuned. We're actually trying to write a review of that right now, just where we are, what we need to find out. Um, and then your second question, I'm sorry, was EEG, right?
Um, yeah, so I mean, there's
Yeah.
the common utility idea with the ventral striatum,
Hmm.
and I think an interesting parallel would be maybe the earlier work on neural synchrony, so whether large groups of people.
Synchrony. Oh, yeah, I like yeah, Moran Cerf did some work on that. Right. So, yeah, so there's an interesting question about synchrony too. Like, is synchrony predicting choice and so forth? And synchrony is more something you look at across individuals, right? And you can get it with EEG signal, you can get it with fMRI signal. Often people will talk about synchrony, but they won't talk about where it's happening. So another question is like, okay, where is this? If synchrony is forecasting something, where is it forecasting? Again, we don't have a lot of data on that, although we're starting to look at it, and it's a very interesting question. For instance, I shouldn't go into it because it'll take another half hour, but we're looking at sort of movie trailers, responses to movie trailers, and whether you can forecast demand for movies out of sample, which of course Alex and others at Rotterdam, Alla, and Martin Boxam have looked at. So we're going to compare actually like the neural forecasting signal that's univariate with the synchrony signal to see if any of those forecasts when and where in the brain it does that. But we haven't figured that out yet. I guess I can give you a teaser to say that the univariate signal seems to forecast, especially early in the trailer. The synchrony signal doesn't, but it tells us something about where the univariate signal might matter. And so that's kind of interesting as well. And now I'm really getting into the weeds. I apologize for all of you who don't think about forecasting all the time.
I have Negin on the list. I hope I pronounced it correctly.
Yes, hello. Yes,
Hi.
that was correct. Thank you for the talk. I was really curious about this idea of using basically the anterior insular activation or the nucleus accumbens activation to then forecast longitudinal riskiness of
Mhm.
participants or the people. And I was wondering when would you think that measurement would be the best? Just like in other studies in the childhood, would you do
Hmm.
it
Wow.
like as a
Yeah.
childhood, or would you do it as an adult? Or what are your thoughts on like the timing of that measurement then?
Yeah. Well, raises developmental questions, which are totally fascinating and unexplored by us. There are these huge data sets, actually, longitudinal data sets, mostly looking at addiction-related phenomenon and collecting some of these measures, including diffusion-weighted imaging and fMRI, even using the mid-task. And so there is an opportunity actually to look at what precedes what. Like if you see some individual difference in these signals, does it then predict that that person may be more at risk, say, for developing addiction or something like that? Um, and so people have started to look at that, and there's some preliminary results on it. But again, it's very difficult because of the not just the difference between, kids and adults, which is very important, but also the quality of the data sets and so forth. So there are all these questions. And then also generalizability across different samples collected in different countries and different cities and so forth. So there are a lot of thorny problems that have arisen from those data sets, which I have to give people a lot of credit for collecting all this data. And I think that it's going to be very interesting going forward to continue to analyze this data. But just full disclosure, all the data I talked about was in people over 18. So these are, people who are like young adults, basically, and probably super healthy young adults. Like these are, people near Stanford or UCLA. And so these are probably not representative of most of the world. And that's who we tend to start with these neuroimaging studies, and then we start to look at other sort of samples and populations. So, and of course, our lab is very interested in addiction, and continues to be that way. But we always figure we're going to try to nail things down in a healthy young adult sample first, and then if we can get something that we can rely on, then try to look at how does that generalize to people of different socioeconomic statuses or ages, or with psychiatric issues. Again, I feel like I'm not answering your questions. I'm just raising more questions here.
No, but it's great food of thought. Thank you.
Sure.
All right. I would also have tons of questions, but I'm afraid we've run out of time. So please give Brian a virtual applause. And thanks so much, Brian, for coming. It was really super fascinating.
Thank you, guys. It was a delight to talk to you. I look forward to doing this in real life at some point.
And just as a teaser, we have our next talk in the Motivated Cognition meetings on February 25th with Michael Treadway, and everybody's cordially invited to join. Thanks so much for coming, and have a nice remaining morning or evening wherever you are.